Good morning. Welcome to my channel Science and Society. Today we are going to talk about plasma membrane. As we know, plasma membrane is a semi-permeable barrier composed of lipid bilayer that separates a cell from its surrounding. Okay, so it's a barrier which segregates a cell from its surroundings. Suppose this is an animal cell. So this outer boundary is the plasma membrane. Similarly, plant cell. It is after cell wall here is plasma membrane. Now we have complete detailed structure of the plasma membrane. We know how different proteins are located, how phospholipids are located. Everything we know about that. But this is not exactly how the picture was. So we will come to about historical perspectives, how different models of the plasma membrane were established, and what were the evidences. Based on those evidences, how different models were portrayed, different models were proposed, and we keep on adding new and new information and new models emerge. And ultimately, current well-accepted model of the plasma membrane is fluid mosaic model. If we go back to the history, in 17th century there was the invention of the light microscope. Under light microscope, when different sections, different parts of the animals or plants were seen, and it was found that they are very delicate structures. Similarly, Stildian and Sichuan. Stildian was a botanist and Sichuan is a zoologist. They concluded simultaneously that all living tissues, they are composed of individual cellular units. Robert Hooke, he looked at a slice or cork or bark of the tissue, and he found this honeycomb-like structure, as we know about these things. Then in 1858, Virchow stated cell theory. According to cell theory, the cell is the basic structural and functional unit of the any living tissue. So, if cell is having this individuality, so there must be a boundary which segregates one cell from another cell, which segregates one cell from its environment. So what exact is the nature of this boundary? We were not sure what is the nature of this boundary. What is its chemical composition, how it is made up of. So a series of elegant experiments in 1890s were performed by Overton. He used almost 500 molecules and he used cells as osmometers. So these molecules, these solutes were allowed to pass through this barrier into the cell, and he found two trends. Small molecules enter cells readily than large molecule. Okay? If a molecule is small, it will easily enter the cell. Second trend is that rate of entry is directly proportional to molecular solubility, organic or non-polar solvents, and inversely proportional to the polar solvents. So, based on this experiment, we can conclude, or our team conclude, that this is somewhat relatively solid barrier. Because why it is a solid barrier? Because it did not allow all the molecules to pass through it. It is water insoluble, or it is non-polar barrier. Because non-polar compounds were allowed to pass through it, so its nature should be non-polar. So this was the first finding where we came to know about this particular barrier. Then, what are the non-polar compounds in the cell? Biochemistry was an era of biochemistry. So biochemists, they isolated different compounds like carbohydrates, lipids, proteins. Among these compounds, lipids were the most non-polar compounds. Okay? And then there were done some other measurements like physical property measurement of cell boundary, like electric resistance, capacitance, and those were in the range of isolated lipids. So lipids as well as this boundary, they have these physical parameters, and their so the chemical composition should be almost similar. Then came Langmuir. He investigated the surface property of a lipid film that was made up of polar groups. What he found that once that lipid film is put into the water, these polar groups, hydrophilic negative groups, they orient themselves towards the water. Okay, so polar groups were orienting towards the water. Okay, based on this Langmuir's observation, Goethals and Grindel, they extended their study. They took RBC, red blood cells. Red blood cells are very simple cells. They have only one membrane, extra membrane, plasma membrane. They don't have any organelles. So there are not any organelle membranes or endomembranes. So endomembrane system is not present in RBCs. These scientists, they took few RBCs. By means of microscopy, they determined their surface area. Okay, how many RBCs they took, what will be the surface area, and from the same number of the RBCs, they extracted phospholipids. Okay, then they found that the lipid surface area was twice needed to cover the surface. Okay, lipids which were extracted, they were twice the surface area. So they proposed a model that this lipid, it is bimolecular. Okay, bimolecular lipid layer is present here. Earlier experiments proposed that the lipid layer is there. Now Goethals and Grindel, they based their observations on RBCs, what is the surface area and how much lipids they could extract. The lipids that were extracted, they were twice the surface area. So they obviously postulated that lipid bilayer like structure should be this plasma membrane. You can say this is the phospholipid. It has phosphate group and it has two fatty acid chains. Similarly, this molecule also phospholipid and two fatty acid chains. They are facing each other. So bimolecular model was proposed by Goethals and Grindel. Then further revisions took place. Further understanding about the cell membrane came into existence. That revised the membrane concept that it is only composed of lipids. So there are not only lipids which are constituting this plasma membrane, there are other molecules may be present here. One of the experiment was starfish egg. Once starfish egg was placed between two cover surfaces, less force was needed to compress than if the starfish membrane was only made up of lipids. Okay. So, and simultaneously another experiment on lipid film. When in lipid film, if we add proteins, its surface tension was getting decreased. So, from these experiments, Davidson and Daniel, they proposed a model called a sandwich model of plasma membrane. Till that time, it was almost everybody was concluding that lipids are the only components of the plasma membrane. But David and Davidson, based on the evidences, what were the evidences? Key starfish egg once put between the cover surface, less force was needed. So obviously it was not lipid, some other thing was also there. And second evidence was lipid film. Once we add proteins into lipid film, its surface tension was getting decreased. So sandwich model was proposed by Davidson and Daniel. What is in sandwich model? In sandwich model, they propose that these proteins, hydrophilic proteins, they will be covering from the outer side and from the inner side. And this hydrophobic zone, phospholipids, they will be embedded or sandwiched between these protein layers. So this was sandwich model or Davidson-Daniels model of plasma membrane. How they based this model? Because they, based on the evidence, they have found that if we will add proteins to the lipid film, its surface tension is getting decreased. So they postulated that sandwich model might be the exact model portraying the nature of the plasma membrane. Then came, it was good model, it was very simple model, sandwich model was very simple model, and it accounted for various properties of the plasma membrane. Okay, then electron microscopy was done on the cells, on the plasma membrane, and it was found in electron microscopy we are using heavy metals. Electron microscopy of the plasma membrane revealed trinomial structure, as if there were three layers. Three layers were present, with two thick regions, dense regions 2.5 nanometers, separated by clear 4 nanometer thickness, like this. It is like a rail track. Okay. So this trinomial model, which was possible because of high technology electron microscopy. In electron microscopy, we are using electron beam for getting the image of particular structure. So this was somewhat supportive to the sandwich model, because sandwich model also suggested upper outer protein layer, middle phospholipid layer, and inner again protein layer. Okay. Then came unit membrane concept of plasma membrane. This concept was proposed by Robertson in 1957. Robertson, he carefully examined preservation of the outer and inner scalar regions, and he proposed that there is a difference in chemical composition of the outer and inner surface. The proteins from the staining regions, EM microscopic images, he could see that the outer region was somewhat different than the inner region. What he postulated that these outer proteins, those are hydrophilic proteins. Then inner we are having this hydrophilic polar groups, non-polar lipid chains, polar heads. Then inner protein layer, it is also hydrophilic, but it is different. You can see here we are saying we are demarcating it by means of this, and here we are demarcating it by zigzag line. That indicates these proteins are different. The proteins which are present outside, this is called asymmetry of the plasma. It is not symmetrical. It is not the same. The things which are outside, they are different. The things which are inside, and it can exactly EM image, trinomial structure. So unit model concept was proposed by Robertson based on the images. Once he observed minutely images, and he found that the outer region was somewhat different than the inner region. Okay. Then there was advance in microscopy, and different biophysical techniques were discovered. That led to the revision of this unit membrane concept. Okay. What were different techniques like freeze fracture techniques? Freeze fracture techniques allow microscopists to visualize particles in the interhydrophobic region of the membrane. This is the hydrophobic membrane of the protein of the membrane. It was found that there were some particles and those particles were hydrophobic. Mostly those particles were proteins. So it was shifted from the earlier unit membrane concept or sandwich model. Similarly, magnetic circular dichroism, it demonstrated that the membrane proteins are global in nature with hydrophilic and hydrophobic regions. Earlier, whether it's the sandwich model or whether it is a unit membrane concept, we are saying the proteins are on the outer side or the inner side, not in between these phospholipids. So, and then there are other techniques also that demonstrate the fluid nature of the plasma membrane. Based on these findings, fluid mosaic model of plasma membrane was postulated, and this is the currently well accepted model of the plasma membrane. According to fluid mosaic model, it is also based on the asymmetrical lipid bilayer. Okay. The proteins or lipids, phospholipids which are present in the outer leaflet and which are present in the inner leaflet, those are different. That means asymmetry is there. But one more thing is that this layer of the protein is not there. Some proteins are embedded within the phospholipids. If proteins are embedded within the phospholipids, those proteins, their nature should be hydrophobic because they have to interact with the phospholipids. What are these? Fatty acids of the phospholipids. So there are two types of proteins according to fluid mosaic model of plasma membrane, there are two types of the proteins. Some proteins which are present towards the periphery, outer side or inner side of this plasma membrane. Those proteins are called peripheral or extrinsic proteins. Okay? And some proteins which are embedded in this lipid bilayer, and those proteins are called integral proteins or intrinsic proteins. These are integral parts of the membrane. Okay? These proteins, they have outer hydrophilic regions, no doubt about that, but they have some regions which are hydrophobic in nature. Those regions are hydrophobic, they are interacting with the fatty acid chain or hydrocarbon chain of these phospholipids. Okay. One more thing is, so this is the phospholipid. It has a polar phosphate group. Phosphate group is present there, and there can be also some other moieties like carbohydrates can be attached there, oligosaccharides can be attached over there. Then there will be a glycerol molecule, and to that glycerol molecule fatty acids are attached. We are having two chains of fatty acids. This phospholipid is oriented inside, it is forming bimolecular layer. Why bimolecular layer? Outer layer is facing, phosphate is facing water. In the cell we have, inside the cell we have water, and outside the cell we have water. So what thing will face the water? Phosphate groups. So phosphate groups are facing the water, and these fatty acid chains, they are hiding. Similarly, phosphate group is facing the water which is present inside the cell, and these fatty acids, they are facing each other. They are having some interaction called as hydrophobic interaction. Okay, they move away from the water. Suppose if we will put two droplets of oil on the surface of a glass, water glass, in a water glass, we put two droplets of oil. What will happen? After some time, those oil droplets, they will coalesce, they will combine together. Why? Because they are not stable, they are not having any interaction with the water. They prefer to stay together. Similarly, these fatty acid chains, they prefer to stick together. They just combine, they just form this hydrophobic interaction. Okay, and there is asymmetry also. Why it is asymmetry? Because outer proteins, if you can say, glycolipids, phospholipids, they have some carbohydrate attachment. Proteins, they have some carbohydrate attachment. But inner, they are not having any carbohydrates. And this carbohydrate region outside is also called glycocalyx. Okay. So there is asymmetry. And one more thing which is present here is fluidity of the membrane. What do you mean by fluidity? This membrane which is present, which is all along the cell. Suppose we are having this cell. This is an animal cell. It has a membrane all along its surface. This membrane is fluid-like. Its nature is fluid-like. What do you mean by fluid-like nature? Fluid-like nature of the membrane. Very good. What do you mean by fluid-like nature of the membrane? If this phospholipid is over there, it can move along the surface. It can change its location from here to here, and this phospholipid can go over here, this protein can go over here. Like if we are having a jug of water and you will place some like plastic thing over it, it will move all along the surface. These proteins, they move, they show movements all along the plane of the surface. So it is behaving like a fluid. Why? What is the nature of this plasma membrane? It is fluid. It is showing fluid-like nature. So it is having fluid-like nature. Why is fluid-like nature important? It is important because a cell has to increase in size. Once cell increases in size, if it was a solid or static, it cannot increase in size. Suppose we have a cell which is small in size, and after some time, this cell, it will keep on adding molecules, keep on adding biomolecules, and it will increase in size. Because of this fluid nature of the plasma membrane, it is keeps on adding these phospholipids and increasing in size. Okay. Similarly, phagocytosis. What is phagocytosis? A cell is capable of eating something, like white blood cells, macrophages. Those cells, they eat bacteria, foreign things. So that phagocytosis is possible because we have outer membrane that is fluid-like in nature. Okay. Isolation of biological membranes. One more extension to this biomembrane is how can we get pure membranes, pure biological membranes? Earlier, biological membranes were isolated by using different techniques. Like it was very difficult to isolate membrane from the plant cell because cell wall contaminations were also occurring here. So it was not known what exactly is the nature of the plasma membrane. Then came the density gradient centrifugation that allowed us to isolate different endomembranes as well as plasma membrane. And one more thing is that the plasma membrane of the red blood cells is the mostly well studied plasma membrane because RBCs, they don't have any other organelles. Okay, so whatever membranes we are getting, whatever things we are getting, those will be from the plasma membrane. What is the function of these peripheral proteins? What are peripheral proteins? Suppose here we have these proteins which are on the outer leaflet or inner leaflet. Those proteins will be called, these proteins over here and these proteins over here. Okay, these are peripheral proteins. They are present in the periphery, either outer leaflet or inner leaflet. Those proteins, you can dislodge them. You can remove those proteins if you increase the salt concentration. How these proteins are over there? Why they are there? Because they are having some ionic interactions. They are forming some bond with the phosphate and some other proteins. So they are staying there because of ionic interaction. If you increase the salt concentration, what will happen? Salt will, suppose we are giving sodium chloride. That salt will give charge. So this protein will be dislodged. Similarly, if you increase the pH or decrease the pH, what will happen? pH increase, that will change the charge of this protein. It will protonate the protein. Okay, once the protein is protonated, its charge will be changed. So maybe it will now show repulsion with the phosphate groups and it will be dislodged. Whatever integral membrane proteins, can you remove those proteins by increasing pH or by increasing the salt concentration? No, because those are part and parcel of the plasma membrane. They are embedded in this phospholipid bilayer. Okay, so these integral membrane proteins, you will dislodge them. You will use detergents. How will detergents dislodge these proteins? Like phospholipid, it has one phosphate moiety and then it has two fatty acid chains. Phosphate moiety is hydrophilic and these fatty acid chains are hydrophobic in nature. Detergent also is amphipathic in nature. It has hydrophilic region and it has hydrophobic region. So detergents, they will interfere with this phospholipid bilayer and they will disrupt this bilayer. So once this lipid bilayer is disrupted, these proteins will be removed. Okay. What is the function of these proteins? We are having peripheral proteins, we are having integral membrane proteins. Their functions can be transporters, they can be ligands. Okay. And one thing to be noted, if we will see once we are extracting the proteins from the membranes, how can we assess whether that protein membrane extracted, endomembrane extracted, whether it is pure or not? We can extract, suppose if it is a plasma membrane, it should be having high content of sodium, potassium, ATPase form. If it has high concentration of sodium, potassium, ATPase form, that means that marker is present here, so this membrane is from the plasma membrane. Okay. Similarly, lysosomes, acid phosphatase is if it is there, so that membrane, that endomembrane should be from the lysosome. Similarly, mitochondria, succinate dehydrogenase, if it is there, it is from mitochondria. Okay, are you getting me? We are having different membranes. We are having plasma membrane, then we are having internal organelles. Those organelles also have membranes. So lysosome it has membrane, nucleus it has nuclear membrane. So how can we assess the membrane we have isolated by what is called density gradient stratification, whether that membrane is pure or not? We will assess its purity if it has high concentration of molecule for that particular organ. Okay, so I think this is all for this lecture. We will talk more in the next lecture.